Researchers elucidate structure of enigmatic chloroplast protein transport machinery. Researchers elucidate structure of enigmatic chloroplast protein transport machinery. You're listening to Technical News Reading, presented by Hakeem Alibokis Alexander on Spreaker, social podcasting presented for World Reading Club in association with UniquilibriuM. This edition's reading focus comes to us from phys.org and is titled Researchers Elucidate Structure of Enigmatic Chloroplast Protein Transport Machinery. It was published 1 hour ago of this recording on February 1st, 2023, phys.org by Li Yuan from the Chinese Academy of Sciences. Researchers elucidate structure of enigmatic chloroplast protein transport machinery. Chloroplasts. There's a featured image here of the chloroplast. The credit is to Pixabay, Creative Commons. Chloroplasts of algae and plants are the cellular engines that convert solar energy into chemical energy through photosynthesis. These organelles, bounded by an envelope with two membranes, contain their own genome whose expression is tightly coordinated with that of the nuclear genome. The majority of chloroplast proteins are encoded by nuclear genes, translated in the cytosol as precursor proteins containing a transit sequence at their amino terminus that serves as the entry ticket into chloroplasts. Protein import into chloroplast is mediated by two membrane protein complexes called TOC and TIC in the outer and inner envelope membrane, respectively. These complexes play a key role in chloroplast biogenesis, in the assembly of the photosynthetic apparatuses, and in various metabolic pathways. The different protein subunits of TOC and TIC have been identified and characterized, and TOC and TIC have been revealed to form a supercomplex together. However, how different proteins of TOC and TIC assemble together to form the channels for protein translocation across the chloroplast envelope membranes is unclear, and the protein translocation pathways within TOC and TIC remain elusive. In a study published online in Nature, Liu Zhengfeng's group at the Institute of Biophysics of the Chinese Academy of Sciences, in collaboration with Professor Jean-David Rochal from the University of Geneva, Switzerland, provided long-awaited insights into the molecular components, three-dimensional organization, and potential protein translocation pathways of the TOC-TIC supercomplex from a single-celled green algae named Chlamydomonas reinhardti. The researchers elucidated the supramolecular architecture of the TOC-TIC supercomplex through cryo-electron microscopy. 13 different protein subunits in this supercomplex were discovered, with the exception of TIC214 encoded by the chloroplast genome. All other subunits are nuclear encoded. They are assembled into the TOC complex in the outer membrane, the intermembrane space complex, ISC, and the TIC complex in the inner membrane. Remarkably, it was found that the largest membrane protein, TIC214, spans the inner membrane, the intermembrane space, and the outer membrane, linking the other protein subunits like a bridge, and most likely also acting as scaffold. The TOC complex in the outer membrane is mainly composed of TOC34, TOC90, and TOC75, flanked on the TOC90 side by the CTAP4, CTAP3 complex. A hybrid barrel-shaped channel is formed by TOC90 and TOC75 on the outer membrane. The channel contains an entrance on the cytosolic side and two exits opening toward the intermembrane space, as well as a lateral gate facing the lipid bilayer. A phytic acid, also known as inositol hexaphosphate, inositol P6 molecule intercalates at the interface between TOC90 and TIC214, stabilizing their assembly like a wedge. The intermembrane space domain of TIC214, TIC100, TIC56, CTAP3, and CTAP5 intertwine with each other to form a tower-like structure connecting TOC with TIC. In the inner membrane, the membrane-embedded domains of TIC214, TIC20, CTAP5, and three small subunits named SIMP1, SIMP2, and SIMP3 form the TIC complex. Four lipid molecules serve to stabilize the assembly of a funnel-like channel located at the interface between TIC214 and TIC20 and prevent the channel from leaking. Based on the structural data, the researchers analyzed in detail the features of the pores inside the TOC and TIC channels. They were able to predict the interactions between the transit peptide and the TIC complex through molecular dynamics simulation. Supported by the observation of two channel pores connected by a surface groove and the previous biochemical and functional reports, multiple pathways were proposed to account for the translocation of the different pre-proteins through the TOC-TIC supercomplex to distinct local chloroplast compartments. You can explore further the mechanism for chloroplast protein translocation and quality control. You've been listening to Technical News Reading, presented by Hakeem Alibokis Alexander on Spreaker, social podcasting, presented for World Reading Club in association with UniquilibriuM. This edition's reading focus comes to us from phys.org and is titled Researchers Elucidate Structure of Enigmatic Chloroplast Protein Transport Machinery. It was published by Li Yuan at the Chinese Academy of Sciences on February 1st, 2023. You can read this article and others like it for yourself by visiting phys.org. That's phys.org. phys.org. Researchers elucidate structure of enigmatic chloroplast protein transport machinery.